Class 12, Chapter 9, Strategies for Enhancement in Food Production, Exemplars MCQs given by NCERT. First one, the chances of contacting bird flu from a properly cooked food above 100 degrees Celsius, chicken and egg, are very high, moderate or none. So here the chances of getting bird flu if the food is properly cooked above 100 degrees Celsius, the chances is none. Okay, moving on to the next one. A group of animals which are related by descent and share many similarities are referred to as breed, race, variety or species. So they are referred to as breed. Okay, breed means a group of animals which are related by descent and share many similarities. Okay, means they are related and they are having many similarities. We call them as breed. They are belonging to the same breed. Okay, moving on to the next one. Inbreeding is carried out in animal husbandry because it increases the vigor, it improves the breed, increases heterozygosity, or it increases homozygosity. So here answer is increases homozygosity because it is about inbreeding. So inbreeding is carried out in animal husbandry to increase homozygous nature. So homozygosity. Fine. Moving on to the next one, Sonalika and Kalyan Sona. They are the varieties of wheat, rice, millet, or is it a variety of tobacco? So here the answer is it is variety of wheat. Sonalika and Kalyan Sona, they both are the varieties of wheat. Which one of the following is not a fungal disease? Rust of wheat, smut of bajra, black rot of crucifers, red rot of sugarcane. Which is not a fungal disease? So here the answer will be black rot of crucifers. Here this black rot of crucifer is a bacterial disease, it is not a fungal disease. Okay, black rot of crucifer is a bacterial disease and it's not fungal. Fungal disease will include rust of wheat. Fine, it include, we call it as brown rust of wheat is a fungal. Smut of bajra is fungal. Okay, red rot of sugarcane is also a fungal disease. Apart from that, there is also one more layer, that is late blight of potato. Late blight of potato is also because of fungal infection. Okay, whereas black rot of crucifer is a bacterial disease. And by virus, which one? Some examples of disease that is caused by virus includes tobacco mosaic and turnip mosaic. Okay, moving on to the next one. In virus infected plants, the meristematic tissues in both apical and axillary bud are free of virus because the dividing cells are virus resistant, meristem have antiviral compounds, the cell division of meristem are faster than the rate of viral multiplication, or is it viruses cannot multiply within the meristem cells? So here the answer will be option C, that is in virus infected plants, the meristematic tissues in both apical and axillary buds, they are free of virus because the cell division of meristems, they are faster than the rate of viral multiplication. So that is the main reason. Okay, why they are free of virus. In the apical bud area where the meristematic tissue is there, in the axillary bud area also we see the division of cells is much faster compared to the rate of viral multiplication. So there they are infection free. Okay, so we cannot assure that the plant is free from disease if we just take a part of the meristematic tissue and check it whether it is infected with virus or not. Okay, because the spread of virus is slower in that particular area. There the cells division is taking place at much higher rate. Moving on to the next one, several South Indian states raise 2 to 3 crops of rice annually. The agronomic feature that makes this possible is because of shorter rice plant, better irrigation facilities, early yielding rice variety, or is it disease resistant rice variety? Here the answer will be early yielding rice variety. Okay, so several South Indian states, they are able to raise 2 to 3 crops of rice every year, okay, annually, and the agronomic feature that makes it possible, why they are able to grow more, 2 3 crops every year, it is because of the high yielding rice variety. Okay, moving on to the next one, which of the following combination would a sugarcane farmer look for in the sugarcane crop? Is it the thick stem, long internodes, high sugarcane content and disease resistant, or it will be thick stem, high sugar content and profuse flowering, thick stem, short internodes, high sugarcane content or disease resistant, or it will be thick stem, low sugar content, disease resistant? Yes, so here the answer will be the first option, in which it is given that farmer would look for a sugarcane crop which has thick stem should be thick, long internode should be there, high sugar content, and also it should be disease resistant. Okay, next one, fungicide and antibiotics are chemicals that enhance yield and disease resistance, kill pathogenic fungi and bacteria, kill all pathogenic microbes, or kill pathogenic bacteria and fungi respectively. So fungi and antibiotics, they are those chemicals that will kill the fungi are there to kill the fungicides are to kill the pathogenic fungi, and antibiotics is to kill bacteria. Okay, so option B. Next one, use of certain chemicals and radiation to change the base sequence of genes of crop plant is termed as recombinant DNA technology, transgenic mechanism, mutation breeding, or gene therapy. So here the correct answer will be option C, that is mutation breeding. Okay, so certain chemicals and radiation to change the base sequence of genes of crop plant is termed as mutation breeding. So when we are changing the base sequence, okay, of the genes of crop plant, we will call that as, and we are allowing the plant to grow further. Okay, so these all are done in order to grow the plant. Okay, so that we call it as mutation breeding. So use of certain chemicals, by using certain chemicals and radiation, we are changing the base sequence of the genes. So only there is a change in the base sequence. Okay, here the answers question main part is this one. If there is just a change in the base sequence of the genes in the crop, then what type of breeding will take place? There will be a mutational breeding or mutation breeding, because we have brought some mutation in that. Okay, now question number 11, the scientific process by which crop plants are enriched with certain desirable nutrients is called crop protection, breeding, biofortification, or bioremediation. Here the answer will be biofortification, option C. Scientific process by which crop plants are enriched with certain desirable nutrients is called biofortification. The term totipotency, it refers to the capacity of cell to generate whole plant, or is it bud to generate whole plant, seed to germinate whole plant, or cell to enlarge in size? Answer will be cells to generate the whole plant. So totipotency is what? It refers to the capacity of a cell to generate the whole plant. Next question. Given below are a few statements regarding the somatic hybridization. So somatic hybridization, what answer it will be? So protoplast of different cells of the same plant are fused or is it protoplast from the cells of different species can be fused? Protoplast. So if we see during somatic hybridization, okay, so protoplast of different cells of the same plant are fused? No, it should be the protoplast from the cells of different species can be fused here. 12th, 13th, just a second. Yes, so from we are not taking protoplast from the same plant, okay, different cells of the same plant, they are from different species, okay. So in somatic hybridization, what is being done, the protoplast from the cells of different species, they are fused, fine, and so this is correct. And treatment of the cells with cellulase and pectinase is also mandatory. Yes, the hybrid protoplast contain character of only one parental protoplast? No, it's a fusion. So how can it be only one parental? No. So here the correct option is option second and third, which we see in option D. Okay. So during a somatic hybridization, the protoplast from the cells of different species can be fused, and the treatment of cell with cellulase to remove the cellulose wall and pectinase to remove the pectin, okay, deposition, it is also mandatory. Fine. So that easily fusion can take place. Fine. Moving on to the next question. An explant is dead plant, part of the plant used in tissue culture, or part of the plant that expresses a specific gene. So yes, here the answer will be question number 14. Answer is option C. It is a part of the plant which we are using in tissue culture, that is called as explant, EX. Okay, it is explant that is being used in the tissue culture. The biggest constraint of plant breeding is availability of desirable gene in the crop and its valid relatives, infrastructure, trained manpower, or transfer of genes from unrelated resources. So here the answer will be option D, that is transfer of genes from the unrelated resources. So that is the biggest constraint of plant breeding, okay, that is the transfer of genes from unrelated resources, okay. Next question, lysine and tryptophan, they are protein, they are non-essential amino acids, essential amino acids, or aromatic and no acids. So here correct answer will be option C, that is they are essential amino acids. Lysine and tryptophan, both of these are as amino acids, and which type? They are essential amino acids. Next one, micropropagation is propagation of microbes in vitro, or is it propagation of plant in vitro? In vitro propagation of plant, this is correct, okay. So here the correct answer will be, we'll see the other two options also, but anyways this is the correct one. Micropropagation is propagation of micropropagation is the propagation of plant in vitro condition. Fine. Other two options given are propagation of cell in vitro, no, and growing plants on smaller scale. Anyways, so there the answer will be, what is micropropagation? Propagation of plant cells, okay, in vitro condition. Protoplast is another name for protoplasm, or is it an animal cell, or is it a plant cell without a cell wall, or a plant cell? Here the answer will be, it is protoplast is a plant cell without a cell wall, okay. Protoplast is a plant cell without a cell wall. To isolate protoplast, one needs pectinase, cellulase, both pectinase and cellulase, or chitinase. So here answer will be both pectinase and cellulase will be required to isolate the protoplast. Next one, which of the following is a marine fish? Rohu, hilsa, katla, or common carp? So here marine fish, Which one is a marine fish? Hilsa. Okay, Hilsa is an example of marine fish living in seawater. Okay, next one, which of the following products of apiculture is used in cosmetic and polishes? Is it honey, oil, wax or royal jelly? So here the correct answer will be wax. Okay, wax is the product of apiculture which is used in cosmetics and also in polishing. Next one, more than 70% of the livestock population is in Denmark, India and China. Answer will be India and China. So more than 70% of the livestock population we find in India and China. Next one, the agriculture sector of India employs 60% of population, 70% of the population, 30% or 62% of the population. So here the answer will be, correct answer is 62% of the population. Okay, it is 62% of population which is there into agriculture sector in India. Remember the percentage, it is 62%. Moving on to the next one, 33% of India's gross domestic product comes from industry or agriculture. Let us see the other two options, export or small scale cottage industry. So 33%, okay, it is asked 33% of India's gross domestic product comes from agriculture. It comes from agriculture. Fine, agriculture is the correct answer. Moving on to the next one. A collection of all the alleles of all the genes of a crop is called as germplasm collection, protoplasm collection, herbarium or somaclonal collection. What it will be called? So here answer will be option A, that is germplasm collection. Fine. So what is germplasm collection? Germplasm collection is collection of all the alleles of all the genes of a crop plant. All the alleles of all the genes present in a crop plant, that is called as germplasm collection. Okay.